So let us see about quantitative character. And its name indicates these are the characters which shows discontinuous variation, while some character shows continuous variations. When characters showing continuous variation are subjected to measurable data like height of the plant, weight of the seeds, color of the seeds, and any other measurable characters, then these characters are called as quantitative characters. So as the name indicates, here we can calculate the number of seeds, weight of seeds, color of the seeds, and any other measurable data. So that's why it is called as quantitative inheritance. Major differences between quantitative and qualitative inheritance. So quantitative and qualitative character shows following major differences. Quantitative characters, these were discovered by Qualitative in 1760 and Yule in 1906. The character shows degree of variations, for example, fruit size, number of seeds, seed shape, weight of seeds, etc. They do not show clear-cut difference in the individuals. They show continuous variation. Phenotype shows a range of spectrum of variation. The characters are controlled by many genes. Here it is important, these characters are controlled by many genes, and characters are concerned with matings in population, and all gene pool is considered. There is more influence of environment on these factors. Analysis gives estimate of parameters such as mean and standard deviation. So these are some of the characters of the quantitative inheritance. Now let us discuss about qualitative characters. These were discovered by Mendel in 1866. According to Mendel, these characters are of definite type, seed shape, flower color, height of the plant, etc. This shows clear-cut difference in individuals and shows discontinuous variation, produce distinct phenotypic classes, that is either tall or short, having purple or yellow flowers. So characters, these are controlled by single gene. So as we know that whatever studies Mendel have done, fortunately he have selected such characters which were located on different chromosomes, and therefore these characters are controlled by single genes, while in earlier case they were controlled by many genes. These characters are concerned with individual mating type, that is specific genes are considered. There is no effect of environment, this is most important, and analysis done by making counts and ratios. So this was the first topic, and I expect that all of you might have understood different factors from this. Now let us see next topic, multiple factor hypothesis. Here we are going to see the action of different genes on the same character. That is, when one character is controlled by several genes and alleles of all genes governing the trait are similar to each other in their action, these effects are called as additive or cumulative effect. This hypothesis is called as multiple factor hypothesis. That is, these are controlled by many genes. This was postulated by Yule in 1906, and experimental evidence for the same was provided by Nielsen and Eel in 1906. To study multiple factor hypothesis, they took a character of seed color in wheat. In case of wheat, dark red color of seeds is a dominant character, while white color is a recessive character. In a cross between two plants, initially they got 3:1, 15:1, and 63:1 ratio. They conclude that the color of the wheat seeds may be governed by 1, 2, or 3 genes. However, on keen observation of seeds, Nelson found that there is marked difference in the intensity of seed color. Therefore, he classified the seeds based on the color intensity, and he found that the seeds can be further divided into 4 categories having ratio 1:4, 6, 8. So this ratio is quite important. This was the first clear-cut demonstration of existence of multiple factors. So let us see the crossing experiment in wheat as follows. The red wheat crossed with white wheat. The red wheat having genotype capital R1, capital R1, capital R2, capital R2, which are dominant characters, and white wheat having genetic combination as a small r1, small r1, small r2, small r2. If we form the gamete of these genotypes, we can get capital R1, capital R2 from red wheat, and small r1, small r2 from white wheat. The F1 generation will be heterozygous in nature. It will have capital R1, small r1, capital R2, small r2, and this will have medium red color. So we have started with red and white wheat, but here the F1 is having medium red color seed. On selfing of this F1, we can get following checkerboard, that is we can see gametes capital R1, capital R2, capital R1, small r2, capital small r1, capital R2, small r1, small r2. Likewise, the same type of gametes will be here in the right corner of the slide. So we can see here, depending upon the number of dominant gametes present in the genotype, here we can get different colored seeds. So in the first case, we can see here the plant is having 4 dominant genes which is having dark red color. The other plants, they are having heterozygous conditions, and as the number of dominant genes gets decreased, slowly we can get faint and faint color, and lastly when all recessive genes are present, we can get white colored seeds. So if we summarize the whole checkerboard in following manner, so we can get dark seed having all 4 gene dominants, its frequency is only 1, and number of alleles, those are the 4, and it is designated as dark red. Then medium dark red, we will get 4 different types of heterozygous plants which are having 3 dominant genes, and these are the medium dark red color. So then medium red color, these are the 6 individuals which are again heterozygous, and the dominant genes are 2. Then the multiple factor hypothesis can be concluded as in crosses showing 15:1, 36:1 ratio, the character is governed by 2 to 3 genes respectively. The allele producing seed color is called as positive allele, and the allele which is not producing any color in seeds, it is called as negative allele. In this case, the positive allele are capital R1, small r2, and recessive alleles are small r1, small r2. The genes do not show dominance, therefore in heterozygous condition, that is capital R1, small r1, we will get intermediate color. Each positive allele has equal effect on seed color. Intensity of seed color is dependent on number of positive alleles of all genes. Thus, from above observation table, it becomes clear that when genotype is having 4 dominant alleles, it shows dark red colored seeds. In heterozygous conditions, as number of positive allele gets decreased, there is decrease in color intensity, and therefore when only single allele is present, we get light red color seeds. Thus, when one character is controlled by several genes and allele of genes governing that trait are similar to each other in their action, these effects are called as additive or cumulative effect. This is called as multiple factor hypothesis. Now let us see the same phenomenon for the ear length in maize. This is also an example of multiple factor hypothesis, and it has been studied in Indian corn. Height of the plant, length of the ear, and productivity depends on multiple factors. East crossed the strain Tom Thumb having short ear size of 2 inch to 8 inch with black Mexican sweet variety having long ear size. So let us see the cross between these 2 plants. So black Mexican sweet, here we can have black Mexican sweet variety which is having ear length 14 to 21 inch, which is crossed with Tom Thumb variety which has got ear length of 2 to 8 inch. So genotype for the black Mexican variety is L1, L1, L2, L2, that is capital all, and for Tom Thumb variety small l1, small l1, small l2, small l2. So if we form the gametes, we can get from black Mexican sweet, we will get L1, L2 as a gamete, and for Tom Thumb variety, we will get small l1, small l2. The F1 generation, it will be heterozygous, having capital L1, capital L2, small l1, small l2. Now in fact, here are only 2 dominant genes are present and 2 recessive genes are present, and there is no dominance of any gene. Therefore, in F1 generation, we get ear length of 19 to 15 inch. So if we select this plant, we can get the checkerboard as same, and here we can see the gametes in the checkerboard, that is capital L1, capital L2, capital L1, small l2, small l1, capital L2, and small l1, small l2. The same is the case about the column in the left side. Same type of gametes. Here, if we cross the gametes with each other, we can find that in first case all 4 genes are dominant, and that plant will produce a ear having 21-inch length. In other cases, depending upon the number of dominant alleles, as the number of dominant allele get decreased, the size of the ear also get decreased, and therefore we can get various length of ears in case of maize. In case of last one, that is the small l1, small l1, small l2, small l2, all genes are recessive, and therefore here we can get the plant which is having ear length 2 inch. So explanation of the checkerboard. In F1 generation, we get range of ear lengths from 9 inch to 15 inch. On selfing F1 generation, we get a variable range of ear length from 2 inch to 21 inch. From this, it is evident that not only original type appears, but there are all inheritable lengths of ear in F2 generation. So other examples of this polygenic inheritance are, they have been also studied in the plants like oat, turnip, gourd, barley, tobacco, primrose, etc. This list serves to show how often this form of inheritance has been met with, and when it is stated that in number of these plants and animals, several characteristics shows this kind of inheritance and its frequency will be apparent. Many but not all the above cases relate to the size, and size is obviously characters towards which many separate parts contributes. So conclusion, thus we can conclude that the length of ear is not governed by a single gene, but it is the result of multiple genes taking active part in determining size of the ear in maize. Due to this, we get a range of ear length right from 2 inch to 21 inch. This has ultimately resulted in the increase in productivity of maize at a large. This is called as multiple hypothesis for ear length in maize. Thank you very much.